Hi, hello and welcome to Dr. Vinita's bio classes. The aim of this channel is to develop an interest in students towards biology. The topic we are going to study today is the pistils, Megasporogenesis and embryo sac. In the last class we had studied about the male reproductive structure called as the Gynoecium, and now today we are going to study about the female reproductive part which is known as the Gynoecium. And the basic structure of Gynoecium is called as the carpel. Carpel is the basic structure of the female reproductive organ. Okay, and the carpel is composed of three parts. It has a long stalk called as the style. It has at the top, at the tip, it has stigma, and at the bottom there is a bulged region called the ovary. So this is the structure of a carpel. So carpel, which is the basic unit of the female reproductive organ, it has three parts: the stigma, style and the ovary. The stigma is the part where the pollen grains come and settle. That is, it is the landing platform for the pollen grains. And there is a long stalk called the style, which allows the passage of the pollen grains into the ovary, wherein the female gametophyte is present. Okay, so now we'll study the structure of ovary. So this carpel can be present in different numbers in different plants. For example, in some plants there will be only one carpel, and in some others there will be more than one carpel. So if it is only one carpel, then it is known as monocarpellary. If it is one carpel, then it is called monocarpellary. And if there are more than one carpel, then it is known as multicarpellary. This multicarpellary. Okay, and this multicarpellary, it can be of two types. I told you this is the basic structure of the carpel. Carpel, if it is available in more than one numbers, then it is known as multicarpellary. And this multicarpellary, it can be of two types if the carpels are fused together, then it is known as, if it is fused together, then it is known as syncarpus. And if the carpels are free, then it is known as apocarpus. Okay, so this is how we name the plants based on the carpels, number of carpels. But we haven't said about this term pistil, right? We have been saying about the carpels. What is pistil and carpel? Are pistil and carpel all the same? Not essentially, because pistil can be one or more carpel. Okay, if only one carpel is there in a plant, we can say that that plant has one pistil. That's not a problem. But if the plant has more than one carpel, then also we say it as a pistil. Okay, so if it is monocarpellary also we call it as a pistil. If it is multicarpellary also we call it as a pistil. Okay, so pistil and carpel is not the same all the time. The ovary, as we said, is the female reproductive organ. Okay, ovary is the female reproductive organ, and inside this ovary is the ovule, which is otherwise known as Megasporogenesis. This Megasporogenesis is otherwise known as ovule, and this ovule is present within the ovary. This ovary has a cavity within it. Suppose this is the ovary. It has a cavity within it called as the ovarian cavity. This ovarian cavity can be otherwise called as locule. Okay, the ovarian cavity is otherwise known as the locule, and inside the locule is the placenta. Inside the locule is the placenta, and from the placenta arises inside the placenta is the ovule or the megasporangium. Okay, so the ovary has ovarian cavity inside it, which has the placenta, and from the placenta arises the megasporangium or the ovule. There are plants which have one ovary, and there are some other plants which have more than one ovary. The plants which have one ovary, for example, the wheat, paddy, mango, etc., are examples of it. And there are plants which have more than one ovary, such as the papaya, orchids, watermelon, etc. The female gametophyte or the embryo sac. The female gametophyte is otherwise known as the embryo sac. The female gametophyte or the embryo sac develops from the megasporangium or ovule. From the megasporangium or the ovule develops the embryo sac or the female gametophyte. The ovule is a very small structure which is attached to the placenta by a structure called as funicle. The ovule is attached to the placenta by this funicle, and the region where this funicle it fuses with the ovum. Okay, when something is attached to something else, there will be a junction where that thing is attached to it, right? We say funicle as the stalk that attaches the ovum to the placenta, right? The region where the ovum fuses with the funicle is called as the hilum. The ovum is drawn somehow like this. Okay, this is the megaspore mother cell or the ovule. Okay, this is the structure of the ovum, and this is the inside this is the ovule. The ovule or the megaspore is inside the ovum. Okay, and this megaspore is surrounded by a tissue, a mass of tissue known as nucellus. This is a mass of tissue, and this serves the purpose similar to that of the tapetal cells in the microsporangium. Okay, what is the function of the tapetal cells? It gives nourishment, right? Similarly, the nucellus also, it's a food reserve. It gives nourishment to the ovum. And there are outer structures which give a protective envelope to the nucellus and the ovule within, and this is called as integuments. So the integuments, here two integuments are there. So these integuments serve as a outer protective covering. Okay, and you also might have noticed that this area doesn't have the integument. Okay, this particular area doesn't have integument, and this area is known as the micropyle. The tip of the nucellus is an open area where integument is absent. That is, it is not covered by the protective envelope, and this part is known as the micropyle. Okay, and this region serves as a entry point for the pollen grains into the female gametophytes. Okay, so this region is known as the micropyle, and we said that this is a tip, and so when there is a tip, there will be a base also. So the opposite region to the micropyle, this region, it is the base, and this region is known as the chalaza. Okay, so this region is called as the chalazal end or chalaza, and this tip portion is known as the micropyle or micropylar end. Okay, so this is how an ovum looks like. So now the ovule that we are talking about, that is, which is in the center of the ovum, it is a megaspore mother cell. Okay, we are not talking about the fully developed female gametophyte. We are going to talk about it, but now this is an ovule, which is a megaspore mother cell. Okay, the megaspore mother cell, what will be the ploidy of a megaspore mother cell? It will be diploid, right? It will have two sets of chromosomes. Now it is diploid in nature. So the ovule or the megaspore mother cell is 2n ploidy. So the ovule that we were talking about, it undergoes certain changes. Okay, it undergoes a process known as Megasporogenesis. Megasporogenesis. If you remember our previous class, we had talked about the microsporogenesis. And what was microsporogenesis? It was the formation of microspore from microspore mother cell by the process of meiosis, right? So here we have to just replace the pollen grains or the microspore with the megaspore. Okay, just that. So the megasporogenesis is the formation of megaspores from megaspore mother cells. Megaspore mother cells are known as MMC. Okay, megaspore mother cells. So the process of formation of megaspores from megaspore mother cells by the process of meiosis is called megasporogenesis. So megaspore mother cells, we already saw that it has 2n ploidy, right? It was diploid. So this undergoes meiosis, and after meiosis we get haploid cells. Okay, so the megaspores formed are haploid in nature. Okay, this megaspores are haploid in nature, and this megaspore now undergoes few changes. So how many megaspores are produced after megasporogenesis? Already said that meiosis, during the process of meiosis, 4 daughter cells will be produced. So 4 megaspores were produced, right? So 4 megaspores were produced, but the 3 out of the 4 megaspores, they degenerate, and only 1 megaspore acts as a functional megaspore. Functional megaspore, or this megaspore becomes the female gametophyte. Okay, so the female gametophyte, again, it has to undergo certain changes. The functional megaspore will undergo certain changes to form the female gametophyte. But the point is, 4 megaspores were produced as a result of meiosis, but 3 out of the 4 megaspore will get degenerated, and this is by a process known as programmed cell death, which is the technical term for programmed cell death is apoptosis. Apoptosis is programmed cell death. The cells is determined to die at this particular time, so it dies. Okay, so the 3 of the 4 megaspores degenerate, and only 1 megaspore is left, and this 1 megaspore is a functional megaspore, and this functional megaspore then undergoes several changes within it to form the female gametophyte. Okay, so now you are clear about megasporogenesis, right? Formation of a single megaspore after megasporogenesis is known as monosporic development. Okay. This process is known as monosporic development. So now we know that the megaspore which is formed, the functional megaspore, let this be the functional megaspore. Okay, so this functional megaspore has one nucleus in it, right? This is the functional megaspore. It has a single nucleus and its ploidy is N. It is haploid, right? This megaspore will now undergo mitosis. But this mitosis is not applicable for the cytoplasm. It is just for the nucleus. The nucleus undergoes karyokinesis, that is the division of the nucleus. There is no cytokinesis happening here. So the cell division doesn't occur, only the nucleus divides. Okay, and the nuclear division is through mitosis. So the first this nucleus divides into two, and two nucleus will be formed. Then again these two nucleus will again undergo mitosis, and now four nucleus will be formed, and then these four nucleus undergo mitosis again, and then like that 8 nucleus will be formed. So now the single megaspore is 8 nucleated. Okay, it has 8 nucleus in it, but the cell is a single cell. It hasn't undergone any mitosis. So cytokinesis didn't occur, only karyokinesis occurred. Okay, there's a karyokinesis and not the cytokinesis. And so now it is an 8 nucleated stage. Okay, now what happens is that the 6 of the nucleus, okay, the 2 nucleus will remain at 2 nucleus, it will remain at the center itself. Okay, 2 nucleus, it will remain the center, and total it was 8 nucleus, no? So 2 nucleus will remain is in this big cell itself, and 3 nucleus will go to one end and 3 other nucleus will go to the other end. So before going to the pores, these nucleus, they develop cell walls. Okay, these nucleus, they develop cell walls around it. So these nucleus, they develop cell walls around it. But the 2 nucleus which are at the center, they remain together. They are called the polar nuclei. Okay, they remain together at the center, and the other 6 develop a cell wall around it. And 3 of the cells, now we can say that it is 7 cells, right? This is the biggest cell, this is the bigger one, this is the large cell, and inside that cell there are 6 other cells. Okay, 6 other cells. This is similar to that of the male gametophyte. We have already seen in the pollen grains. The pollen grain, it undergoes mitosis, and then it forms the vegetative cell and the generative cell, right? And then it becomes 2-celled, and 60% of them will be 2-celled, and the rest of the 40%, they again undergo the generative cell, again it undergoes mitosis, and it becomes 3-celled. We had seen that. So here also the main cell is there. Other than that, the nucleus, it undergoes the mitosis, and the cell wall is formed around them, and these cells are within the larger cell. Okay, and now these 3 cells will go to the micropylar end. In the picture we saw that this was the micropylar end and this is the chalazal end, right? So one area is the micropylar area and the other is the chalazal area. So the 3 cells here, they move towards the micropylar end. Okay, these 3 cells will move to the micropylar end, and these 3 will move towards the chalazal end. Okay, 3 cells will move to the micropylar end and 3 will move to the chalazal end. And the 3 cells at the chalazal end are known as antipodals. Okay, these are called as antipodals. And the 3 cells which go to the micropylar end, 2 of them are called synergid cells and 1 will be the egg cell. Okay, these cells are called the egg apparatus. So these 3 cells are called the antipodals and these 3 cells form the egg apparatus. And in the egg apparatus, 2 cells are called the synergid cells and 1 is called the egg cell. And the synergid cell, they have some specialized structures and that is for helping the male gamete to enter into these cells. Okay, so this specialized regions in the synergid cells at the micropylar end is called the filiform apparatus. So this is how the female gametophyte looks like. Now we have the female gametophyte which has 2 nuclei which are the polar nuclei which is placed at the center of the cell, and there are 3 cells at the micropylar end which form the egg apparatus and 3 at the chalazal end which form the antipodals. Okay, so now the female gametophyte, the fully developed female gametophyte has 7 cells and they are 8 nucleated, right? This bigger cell has 2 nuclei which is placed at the center, polar nuclei. Okay, so this is how the structure of the female gametophyte looks like. So now we have studied about the pistil, and the embryo sac or the female gametophyte. Thank you.